Good afternoon, Naveen sir.
Yeah, good afternoon, sir. Good afternoon.
Good afternoon, Sanjay sir.
Hello, good afternoon.
Good afternoon, sir. Please wait. We'll be starting by 2:30, sir.
Yes, sure, no problem. So I can load my first slides, or later?
Later, sir, because you'll be introduced to the audience first,
Okay,
and then
sure.
after that you can load, sir.
Sure, no problem.
Good afternoon, all the participants. Good afternoon, sir.
Good afternoon.
Hello, good afternoon, sir. Sanjay
Good
here.
afternoon, Banerjee, good afternoon. So, is there any problem with the net now? It's okay.
No. Now, fine. Everything is fine.
You are coming to the office or from office only now?
No, I'm actually from office. Yeah, I'm presenting from my office.
Okay. Because
So
I
I
was
having a
told that
good.
yeah,
Yeah.
I was told that there is some problem with the net connection there in the institute all over.
Sir, yesterday it was very bad, and today I talked to my IT people. Please give me a good connection. I having a presentation, so I hope they'll give a good support.
Okay.
Good afternoon, sir.
Good afternoon. Good afternoon. Yeah.
Good afternoon, Dr. Sanjay. Good afternoon, sir.
Hello, good afternoon.
Sanjay sir, he's our principal, Dr. Naveen Mohan.
Oh, yeah. Hi, Dr. Naveen.
Hi, sir.
Yes. I saw you three people are very important for this conference. Professor Unni, Dr. Nobin, and Dr. Finos. So you are the the three key players for the whole 10 days conference.
Thank
And
you.
Thank you, sir.
and the way you have like organized, it's not easy for 10 days continue one conference. Generally people finish in 2 days, like 3-4
Yeah.
lecture every day, and keep your spirit on until continuously 10 days, not easy.
Thank you.
Thank you, sir.
We got a good speakers and representing different institutions. That is also, and we all got very hot topics, very recent topics like Devdatta, Bhattacharya gave a very interesting topic on COVID. Entire, very clear picture, he gave. As of today, what is happening and what is going to be in future, everything he mentioned.
Yes, sir. So I think you choose very people from
Yeah.
different part of India
Different parts.
and who are working at different good topics.
Yes,
Like
yes.
kind of expertise you have chosen. They are completely different background and
Yes.
really good topics.
They all are research minded. And what I found is that is very important. As on today, you have to have a research background people working very deeply and different sectors. Suppose what is happening in ICMR, what is happening in hyper government research, what is happening in CSIR, what is happening in IITs, what is happening in state university, what is happening in state research institution. So the faculties of college also will get in Kerala, they will get an idea what is happening outside, because a college teacher faculty will say, research facilities are problem and all sort of things, how can do the academic and all. So this is also an example how they are doing it.
Exactly.
How they bring the project. So that is a good message. And then today is the senior, the UG and the PG faculties are the next days, students are the next days faculties.
Exactly.
So they also get a message.
Yes.
Yes. Yes, Finos, you can go ahead.
Yes, sir. Okay. A very good afternoon to all of you. And today, finally, we are on to our 10th scientific session. And today we have Dr. Sanjay K. Banerjee, who is the Associate Professor, National Institute of Pharmaceutical Education and Research, Guwahati, Assam, with us for a speech. I welcome you, sir.
Thank you. Thank you,
Next,
Dr.
thank you, sir. Next, I'd like to welcome Dr. B.G. Unni, who is the advisor as well as Director of Academics and Research to this event. Welcome, sir.
Thank you, Dr. Finos. Thank you.
Next, I'd like to welcome Dr. Naveen Mohan, who is our principal and also the convener of this program to this event. Welcome, sir.
Yeah, thank you, Dr. Finos.
Next, I'd like to welcome all the participants from all over India to this event. And I'd like to welcome Ms. Ranjisha VP, Assistant Professor in Biotechnology, to introduce the guest to the audience.
Good afternoon, everyone, and welcome to today's section. It's my honor to introduce our today's presenter, Dr. Sanjay K. Banerjee, sir, Associate Professor, National Institute of Pharmaceutical Education and Research, Guwahati, Assam. Sir completed his PhD from AIIMS in 2004. Dr. Sanjay K. Banerjee, sir, also worked in THSTI, CSIR, IICT, University of Pittsburgh, New York State University. Sir have about 94 publications, 6 book chapters, 14 review papers, and 8 patencies. Sir won various awards such as Fast Track Young Scientist Award, American Heart Association Fellowship, Best Postdoc Award from University of Pittsburgh, Travel Scholarship from Keystone Conference, Postdoc Fellowship from Hillgrove Foundation, Ramalinga Swamy Fellowship, Professor Shailendra K. Vajpayee Award from Indian Society of Hypertension. Sir is a member of various prestigious societies and committees, IPS, ISHR, IACS, AHA, DBT, DHR, European Association for the Study of Diabetes. The core area of research it is drug discovery and translational research in cardiometabolic disorder. Dr. Sanjay K. Banerjee, sir, the key research findings such as SGLTI, a novel glucose transporter in heart, sirtuin as a therapeutic target for diabetic cardiomyopathy, role of vitamin D deficiency in cardiometabolic disorder, identification of one novel sulfur metabolites from garlic for heart failure, serum biomarker identification from diabetes patient at high risk of CAD, TLR4 as novel target for cardiac fibrosis and heart failure. So we are really blessed, sir, and we are waiting to hear from you, sir. And once again, welcome, sir, for our 10th National Webinar Conference 2020. And welcome all other participants. Thank you.
Thank you, Ms.
Thank you,
Ranjisha.
Dr. Ranju. Thank you, Dr. Anju. I think it's a nice introduction. And
Thank you,
I'm
sir.
now putting my slides so that you can see it.
Sure, sir.
So let me know if you can see my first slides.
Yes, sir, you can.
Yes, sir.
Yeah,
Yeah,
we can see. You can see.
You can see. Okay. First, I'd like to thank Professor BG Uni. I'm here because of him. And, he knows me last few years and we are in a one project, TBT project. And then also thank to Dr. Naveen Mohan, who is the convener of this conference, Dr. Finos, the coordinator, and thanks for inviting me this national e-conference on current trend in life sciences. And thanks to the whole Society of James Arts and Science College for organizing such a important and timely this conference. So I'm last 10-15 years I'm working on cardiometabolic disease, try to understand the molecular mechanism of the disease, different risk factors, and how we can go for therapeutic interventions. So today I'm going to talk very important topics. You all aware that vitamin D. So how vitamin D deficiency linked to the cardiometabolic syndrome. So I'll talk about a study from bed to bench. So you all know, that vitamin D is a very old vitamin, and it was almost 100 years ago. Scientists discovered vitamin D. So the first, when we realized about vitamin D, we 1999 the ricket, that disease was linked to the deficiency of a dietary factor and lack of sunlight. And 1922 we realized, Elmer McCullum and Margaret Davis, they discovered a substance in cod liver oil which might improve the ricket conditions. And then another 13 years, 1935, the structure of vitamin D, that came, and it was a structure of kind of steroidal structure. So today my talk mostly on vitamin D deficiency. So this is a vitamin D deficiency world curve, and you can look actually the places where is a dark red color, mostly Southeast Asian part of the world, showing they are basically 60 to 69% of the population are vitamin D deficiency. So although we are having enough sunlight, we having is most of the exposure of the sunlight compared to other part of the countries, other part of the world, but still we got a good number of population in India got vitamin D deficiency. So now I like to tell you what is the different ranges in the blood vitamin D concentration. You can tell, yes, I have vitamin D deficiency. So vitamin D deficiency, when we talk about, we generally measure 25 hydroxy D. And if your body level have vitamin D more than 30 nanogram per ml, so you are normal. And if you are between 20 insufficiency, which is below 20, yes, you are vitamin D deficiency. So you can take supplementation of vitamin D. And if you're below 10, it's a severe deficiency. And when you're having a below 10, you'll definitely get kind of symptoms like very weakness, a lot of other symptoms, and then you will come to know that you having a vitamin D deficiency. So researcher concluded that having low levels of vitamin D, like if it is 17.8 nanogram per ml, that was independently associated with an increase in all cause of mortality in the general population. So now, what is the different reasons of vitamin D deficiency? As I know, there are different, kind of students are there who might know what are the different kind of reasons for vitamin D deficiency. So lack of sun exposure, that is the first reason that most of the Indians nowadays, we hate sunlight, we don't go, and that is the first reasons we get vitamin D deficiency. Second reason is less intake of food naturally containing vitamin D. Third is aging and obesity. As you are getting aged, your vitamin D synthesis inside of the body that decreases. And fat malabsorptions, that is linked to vitamin D deficiency. Dark skin, if you're having more pigmentation in your skin, so your sunlight not able to produce a lot of vitamin D. Extreme anti-seizure medication, certain kind of medication, if you take, then they decrease vitamin D. And also you having a liver and kidney disease, that also decrease vitamin D. So I'll tell about why. Okay. So what are the common symptoms? How you know that you have vitamin D deficiency? The first thing, you get a fatigue, you get a muscle weakness and pain. So this is very common. Like I've seen a lot of my relatives, friends, when they actually complain about muscle pain and weakness, I ask them to go for test vitamin D, and they come up with a tell that yes, they are vitamin D deficiency. So when they took vitamin D supplements in few weeks, they they are all muscle weakness gone. So it is, directly linked to, muscle weakness and fatigue. You can get muscle cramps, then chronic joint pain, weight gain, high blood pressure, restless sleep, poor concentration, headache, bladder problem, and constipation or diarrhea. So these are the different common symptoms. If you have any of them or some of them together, you should definitely check for vitamin D. Then what you'll do, you should definitely take foods which are rich in vitamin D, like fish oil, beef liver, milk, yogurt, and cheeses. So sometimes I feel good when I see this awareness is there in media also. Sometimes in several newspapers, they give the message that 65 to 70% Indians are vitamin D deficient, and they written very well why we should concern, why should we worry, and they written few disease name, that osteoporosis, ricket, cardiovascular disease, diabetes, and cancer, even infections. These are the important diseases which are linked to vitamin D deficiency. So there are these are the different human studies, very big human studies happening last several years, and some of the studies participants are very high, like 15,000. Some of them are almost 1 lakh. So those studies telling that vitamin D deficiency or low level of serum 25 hydroxy D are linked to different kind of diseases like CVD, cardiovascular disease, like hypertension, diabetes mellitus, obesity, and also high serum triglyceride levels. So there are several kind of literatures are there, and several studies has been done. So there is no confusion, or there is we know that vitamin D deficiency is linked to cardiometabolic disease and so many other diseases. So one of the study in India that published in 2016, and they looked at so CAD is a coronary artery disease, like when you're having a blockage in your blood vessels, like when your blood vessels like coronary artery having 50 to 70% blockage. So you have we found that 66.7% population out of those have vitamin D deficiency. Now, when the people having a 70 to 90% blockage of their blood vessels, they are, and in that population, 83% are vitamin D deficiency. And when you're having blockage almost 90%, and that time you'll get a heart attack or a lot of other problems. So that also, those population also 83%, they are vitamin D deficiency. That means that if you having a, blockage in the blood vessels, it could, that is linked to vitamin D deficiency very strongly. But this study was not, very much conclusive, because in Indian population, this study shown in the control population who don't have any blockage in their blood vessels or coronary arteries, 80% normal patients, they are also vitamin D deficiency. So it was not very conclusive data. So another important disease which is very linked to vitamin D deficiency is type 1 diabetes. In Finland, actually 10 years ago, there are a lot of actually children, they get type 1 diabetes. So this is an autoimmune disease. Their pancreas damage and due to autoimmune the reaction. So now they have done one study with a vitamin D supplementation, and they found that if we give a vitamin D supplementation, the incidence of type 1 diabetes that reduce. There are also some studies with which is linked to type 2 diabetes and also metabolic syndromes. And most of the cases, they that link to the receptor called VDR, vitamin D receptor. When it is activation happening through vitamin D or 25 hydroxy D. They reduce the insulin responsiveness, or actually it increase insulin sensitiveness actually when vitamin D bind to the VDR. That they reduce insulin resistance in the patients. So before I talk about some of my research data, human and then animal, I'll tell you ki when you talk about vitamin D, there are two type of vitamin D is there, vitamin D2 and vitamin D3. Vitamin D2 mostly source are plant and mushroom, and vitamin D3 source are animal, eggs, and fatty fish. So, but when this vitamin D2 and D3 are formed in the body, they are not active form. They are in the blood circulation, but they have to go through two different changes in their structure. The first change is happening in liver. So what happened? There's one hydroxylation happen in 25% with the enzymes called 25 hydroxylase enzymes, or called CYP27A1. And once they form the 25 hydroxy vitamin D2 or D3, it basically circulate to the kidney. Or another enzymes present is called 1 alpha hydroxylase, or name is CYP27B1, and that makes another hydroxylation of the vitamin D. So ultimately they make 125 dihydroxy D3 or 125 dihydroxy D2. These two are very active form of vitamin D. Until unless you get this active form of vitamin D, so even in your blood has a enough or sufficient amount of vitamin D2, D3, there is no point of that level. So this active form of vitamin D, that 125 dihydroxy vitamin D, they bind to the VDR receptor, like vitamin D receptor, and they actually alter a lot of gene expression which are related to the cardiometabolic disease. So important part is that's why when you having a problem in liver and kidney, if you're having a damage in liver and kidney, so those enzyme expression that also change. So if you don't having enough enzymes in liver and kidney, that 25 hydroxylase enzymes or 1 alpha hydroxylase, so definitely vitamin D formation will be the active form of vitamin D will not form. So ultimately you not see the effect of vitamin D. That's why it is very important when you are taking vitamin D, you have to know that don't having a liver or kidney problem. If you having a liver and kidney problem, vitamin D supplementation may not work for you. So that time you can take a direct active form of vitamin D, which will be more helpful. So once the active form of vitamin D form 125 hydroxy D, so it play a very important role on calcium homeostasis, muscle health, bone health, blood pressure regulation, cardiovascular health, neurodevelopment, immunomodulations, and also play important role in immunomodulation like prevention of autoimmune disease, control of invading pathogenesis, and several pathways that regulate cell growth and differentiation. So now when you think about cardiometabolic disease, so here you can see there are several signaling pathways are linked to cardiometabolic disease. Some of them are inflammation. If you have vitamin D deficiency, that enhance inflammation, macrophage infiltration, increase leukocyte and endothelial adhesion, and that activate atherosclerosis. Other way, you vitamin D deficiency causes increased insulin resistance, and that directly linked to the diabetes, and that causing a lot of change in your blood pressure. You got a left ventricular hypertrophy, the heart size increases, and also cardiovascular event increases, like you can get heart attack also. So overall, you can get more morbidity and mortality due to cardiovascular incidents. So these are some of the studies whatever done before, and they come up with some conclusion: vitamin D deficiency directly linked to cardiometabolic disease. So when we plan to do this study, as I said, in India there is not a lot of study with the patients. So we thought we should do one human study to confirm that yes, vitamin D deficiency is directly linked to type 2 diabetes or coronary artery disease, or together. Most of the 80% of type 2 diabetic patients, they get the cardiovascular complications. So type 2 diabetes patients, there are more risk factor for coronary artery disease, which means your blockage of the coronary artery, which gives heart attack. So we wanted to see what is the status in Indian patients if you take a type 2 diabetic patients or coronary artery, or you combine the two disease, what happened to them. So we started the study in Hyderabad when I was in CSR Institute, and we work with MedCity hospital, and we get the ethical approval, and we started the work with Dr. Mohan. So when we actually started to recruit patients, so we having some inclusion criteria, because you have to know what we define the control and healthy groups. So they should not having they should not have any prior history of type 2 diabetes, hypertension, coronary artery disease, or any other like cardiovascular disease. So they are not taking any medication for any chronic medical conditions. Their fasting blood glucose level and glycated hemoglobin and blood chemistry will be normal. When you talk about type 2 diabetes, the glycated hemoglobin level should be more than 6.5% as per American Diabetic Association guidelines. And they should having a proven history of type 2 diabetes, but no other complication. So we remember type 2 diabetes, getting patients with no complications is very difficult. So early stage of type 2 diabetes, they are very pure form of type 2 diabetes. They don't get any other complication, like they don't have any kidney problem, they don't have any cardiovascular complication, or they don't have any retinopathy or neuropathy. So group 3, which is the coronary artery disease, that we define as narrowing or blockage of one or more epicardial coronary artery with greater than 25% stenosis. Your blockage will be at least 25% in coronary artery, and that will be done by angiography techniques or diagnosed by cardiologist. And this group had no prior history of type 2 diabetes. The fourth group we kept it, that is a mixed of type 2 diabetes and also CAD. Those patients should have all the criteria that match with type 2 diabetes and all the criteria that match to CAD. So what we did, we take the blood from all patients, and then we measure 6 different vitamin D metabolites, like D3. Then, as I said, the first metabolite is from 25 hydroxy D3, and the last active form is metabolism 125 dihydroxy D3. Similarly, D2, then 25 hydroxy D2 and 125 dihydroxy D2. So as we choose 6 different parameters, and it was not easy to measure, so we took help from LCMS. And as I was in CSR IICT, this is a very good chemist was there, and they helped me to standardize all 6 parameters, and we have measured all the patients, all 6 metabolites of vitamin D. Now, when some of the my data, when I talk about total 25 hydroxy D, that means this is a combination of 25 hydroxy D2 and 25 hydroxy D3 together. And when you talk about total 125 dihydroxy D, that means again it is a combination of 125 dihydroxy D2 and 125 dihydroxy D3. So in general, if you look at how, if you see the x-axis and y-axis, x-axis basically increasing the glycated hemoglobin level, that is HbA1c. As the percentage increases, they are more actually diabetic patients. So if it cross 6.5, they are diabetic. So as they are getting more towards 10, 12, or 14, their blood level 25 hydroxy D is lower. So the curve clearly showing there is an association of 25 hydroxy D level in the blood, as well as their increased HbA1c level. Like similarly, we also correlate with fasting blood glucose level. So if you are cross 95 or 100 or 110 maximum, then you will be kind of diabetic patients. So if you cross that fasting blood glucose level more and more, so your 25 hydroxy D level showing lower. That both actually data showing that it is directly linked with the diabetes. So this data actually, when I try to put together all the data and then try to see how actually the data looks. So look in the first graph and the last 3 data, that when the patients have less than 20 nanogram per ml, the 25 hydroxy D. So if 10% populations of type 2 diabetes, they're having a lower than 20 nanogram per ml vitamin D level, like 25 hydroxy D. If you have CAD patients, like 25% of the CAD patient, they're having a lower 20 nanogram. But if it is a type 2 diabetes with CAT, if you are having a complexity of type 2 diabetes with a cardiovascular complication, so 47% patient, they are showing less than 20 nanogram per ml. Now, so the next graph in the right side, and look that now this is 125 hydroxy D. So very few lab basically measure. This is not easy to measure because their level is to identify them, and the level is too low. Only method is LCMS. You can do that. Although there are some actually kit recently came where you can measure the active form of vitamin D, this 125 dihydroxy D. So if you look the less than 11 nanogram per ml, blood, the active form of vitamin D, showing 27%, even the healthy patients, they having a lower than 11 nanogram per ml. If it is a 41%, so then it is they are like type 2 diabetic patients. If it is a CAT, that 50% patients they having a lower that 11 nanogram per ml, and 80% of the type 2 diabetes with CAT patients they having a lower level of dihydroxy vitamin D. So it's very clearly shown that as you are going to type 2 diabetes, CAD, and together the complexity of the disease, your vitamin D level is low. So there's a possibility that as the disease complexity increases, your body not forming the active form of vitamin D, that is possibility is there. Or possibility is that because you're having a low level of vitamin D, that causing the problem. So because 80% of the patients, they're having a lower level of vitamin D, and they basically got this complexity. So we don't know is in this time point whether this is a cause or effect, whether vitamin D deficiency causing the complexity, or because they are in a complex state of the disease, so their vitamin D active form is not forming properly. So look the data here. If you have a control and then CAT and type 2 diabetes CAT, it's slowly, and if you go to more complexity, that total 25 hydroxy D as well as total 125 dihydroxy D level will be slowly decreases as the going to the complexity state. Like similarly, if you go to control, then type 2 diabetes and then type 2 diabetes with CAT, at the same way it's showing that it is the total 25 hydroxy D and active form of vitamin D, there decreases as you going to the complexity of the disease. So we are able to publish the data in scientific reports, so it telling that conclude that okay, lower total 25 hydroxy D level is more associated with type 2 diabetes, while lower active form of vitamin D 125 hydroxy D is associated with the complexity of the disease like type 2 diabetes with CAD. When we adjusted all other established risk factors like age, female, smoking history, alcoholic history, and family history. But at this time when we did this, we did know the cause and effect of this relationship. So when I was presenting, the questions coming, okay, how do you know vitamin D deficiency causing this problem? Or maybe the vitamin D deficiency happening because they have the problem. So we basically thought we'll do some animal study, but at the same time we also done some other human study, which is hypertensive disorder in pregnancy and vitamin D deficiency. So there are healthy pregnancy state, then gestational hypertension, like who are pregnant with a gestational hypertension, then it is getting more actually like critical case is called preeclampsia. So they're having a more higher level of blood pressures. So this is preeclampsia, and then eclampsia, EC. This is another level when patient basically died during pregnancy time. So as you are getting more severe form of hypertension, and pregnancy level women, they having a lower level of 25 hydroxy D level as well as one lower level of 125 dihydroxy D level. So we able to correlate that hypertension in pregnancy also associated with vitamin D deficiency. So we also published that work in Frontiers in Immunology, where we show that how cytokines and chemokines play important role in causing this complication along with the vitamin D deficiency. So there are few studies saying that if you go to the sunlight, this study saying that if you go to UV light, UVB radiation 3 times in a week for 3 months, so 25 hydroxy vitamin D level will be increased approximately 180%, and also shown that blood pressure will become normal. So there's a direct link, like who had the very mild stage of hypertension. If your vitamin D level is low, if you go to sunlight and continuously for few months, you will definitely see improvement. So those study published in long time back 1998 in Lancet. Then also a study that vitamin D linked to the cardiac function and heart failure. and they found that taking vitamin D supplementation may lead to beneficial reverse remodeling. So this study is showing that vitamin D supplementation can help for improvement of cardiac function. So now we thought now we go to from human study to animal study to really find that vitamin D deficiency that cause all the problem. So when we do translational study, as I said, my title was from bed to bench. So what happened? Like we have started the work from patients. So we realized the problem is exist, and we found the association with diabetes and coronary artery disease with vitamin D deficiency. Now we have to go to the bench to find out it is really happening in animal level. Once we find in animal level, and you can think about then treatment, and you can again go to the bed. So this is a cycle. So here I'm talking about bench to bed, but so many times we started work in the bench level with the animals, and then the findings we try to correlate to the humans. So as I said, the study basically to find a cause effect relationship, and so we keep two main objectives for this animal study: to find out the role of vitamin D deficiency on insulin resistance and associated cardiac complications, and second objective to find out if vitamin D receptor activator pericalcital can attenuate insulin resistance and cardiac dysfunction in high fat, high fructose fat rat model. So first objective, purely we want to see that vitamin D deficiency causing a cardiovascular complication. So this is very simple study, but second one, we choose VDR agonist. Why? Because as we know, sometimes, vitamin D, if you are giving supplement and because of the kidney and liver problem, active form of vitamin D is not forming. So I need to give a activator of VDR, vitamin D receptor, where the active form of vitamin D bind. So we find a drug, it's called pericalcitol, which is mostly used for kidney failure patients. And so that drug basically works through VDR. So we use this drug, which is structure also very similar to vitamin D2 level structure. So we use these drugs to the animal, see that if you directly activate VDR, we can see any improvement on cardiac dysfunction as well as insulin resistance in the high fat, high fructose model. So the first study, experiment 1, where we took 3 groups: control diet, where they are getting everything normal. Second group, vitamin D deficiency diet. Here actually we made the food where we have everything except we have not given vitamin D in this food. And the third one, just to compare that vitamin D deficiency diet causing insulin resistance or other complication, which is very similar to high fat, high fructose diet. Because this is a very well known that if you give a high fat, high fructose like chronically to animal, they develop metabolic syndrome, they develop insulin resistance, hyperglycemia, and even cardiac dysfunction. So I want to compare here how vitamin D deficiency diet, causing metabolic or cardiometabolic problem, and how much it is similar to high fat, high fructose diet. So the first study we have measured the blood glucose level. You can look into the left side in the top. So we measured the whether after giving high fat, high fructose 20 weeks, and also control minus, that means vitamin D deficiency groups. So you can see that we have seen the increase of blood glucose level only in the high fat, high fructose group, but not found in vitamin D deficiency food. But when we look into serum insulin level, surprisingly, that vitamin D deficiency diet, which is control minus, showing a very high level of insulin level. And also similarly found high level in high fat, high fructose. Generally, type 2 diabetic patient in their early stage or pre-diabetic stage, they show high level of insulin level. And now there's a parameter, it's called HOMO-IR. It is a basically multiplication of fasting blood glucose level and insulin, then divided by 405, which give you insulin resistance. So you can see how much insulin resistance is there in this animal. When I did this HOMO-IR calculation, looks that both vitamin D deficiency diet as well as high fat, high fructose diet showing very similar data. They're telling you that vitamin D deficiency alone can do the similar insulin resistance as does by high fat, high fructose diet. Now another way you can look insulin resistance is called intraperitoneal glucose tolerance test, where we do give a 2 gram per kg intraperitoneal dose of glucose. If you give a high dose of glucose, there will definitely spike up the blood glucose level. you can see the highest level in 15 or to 30 minutes, and with time it will come back to the normal. So around 120 minutes, it generally come back to the normal. You can see here actually the control, they are having a high blood glucose level, but very little change is below 200, and with time it come back to the normal. But the both control minus like vitamin D deficiency diet and high fat, high fructose, they show the similar curve. So it telling us, yes, vitamin D causes same problem as high fat, high fructose diet. Then we measure serum triglyceride level and serum HDL level. So serum triglyceride level was again, it was high in both vitamin D deficiency diet and high fat, high fructose. When you measure HDL level, surprisingly, high density lipoprotein, which is a protective lipoprotein molecule, which should be, always in a, good level, like in human per deciliter. This animal model, we saw that they had increased the age level, same as high fat, high fructose diet. Then we measure that systolic and diastolic blood pressure as well as mean arterial pressure. Here also surprisingly, we've seen that vitamin D deficiency, although it was not as high as high fat, high fructose, but definitely there's a trend is there. And systolic and diastolic blood pressure levels higher in vitamin D deficiency diet as well as mean arterial pressure. Then we plan to look cardiac function. So we use echocardiography on the animals, where we see that how the heart is functioning. So whether vitamin D deficiency have any or cause any problem in cardiac function. So this is the graph, is a 2D echo. Like the curve is showing that when heart is contract, so these two curves, they are coming near to each other. When they are relaxed heart, so they're going towards the diameter of this distance will be much more. So that way, heart contracts and relax. So we measure the distance and that tell us whether they have any problem in the contraction. Also, we can measure that, the distance, the green color, actually that telling that posterior wall thickness, whether, if the muscles have any increase of the thickness, that are also we can know by doing this echocardiographic study. So here you can look that intraventricular septal, thickness that was increased only in high fat, high fructose group, not in vitamin D deficiency diet. But when you look left ventricular posterior wall thickness, so generally thickness increases when heart got difficulties to pumping. So when we having a blood pressure, so heart has to give extra pressure to pump. So definitely it is directly linked to your blood pressure. So you can look that, and when heart had to pump, more, with a more pressure, so definitely the muscle thickness increases. So here we can see in the left ventricular posterior wall thickness in the right side graph, in both vitamin D deficiency diet as well as high fat, high fructose diet, there's an increase of, this thickness. So which telling that is cardiac, the structure that change because of vitamin D deficiency. Then we measure some other parameter, left ventricular internal diameter, which is happening during diastole, like when heart relax. Another is left ventricular internal diameter. This is systole, S means systole. In the right side shows that left ventricular internal diameter systole only change in high fat, high fructose diet, not in vitamin D deficiency diet. So this is two important parameters. Most of the cases when you go to a hospital and we look cardiac function, doctors, they prefer to measure these two parameters. One is called fractional shortening, another called ejection fraction. So the fractional shortening, where basically how much contraction and relaxation happening in the heart. So this is a kind of ratio, or so you can see in both vitamin D deficiency diet in con minus as well as high fat, high fructose diet, showing there's a decrease of fractional shortening. At the same time, in ejection fraction, you can see there is a decrease. So ejection fraction basically every time's heart pump, how much blood it throws to the body. So it's its capacity of throwing blood to the body. So yes, there is a decrease of the ejection fraction in both vitamin D deficiency diet as well high fat, high fructose diet. So these two parameters clearly tells that if you having a vitamin D deficiency, your cardiac function is also affected. So now We want to look actually in some of the molecular study in this heart. So one of the important parameters when we talk about insulin resistance is phospho-AKT level. So when insulin secretes in the blood, they activate phospho-AKT. Phospho-AKT, they through signaling pathway, downstream signaling pathway, it activates GLUT4 to translocate in the membrane and it enhance glucose uptake. But during insulin resistance, phospho-AKT will not form much, so GLUT4 translocation is also not going to happen. So that way we can get insulin resistance. So here we wanted to see that cardiac function that we have seen some deficiency of cardiac function, whether this is happening due to insulin resistance or not. So we measure phospho-AKT level in control group, vitamin D deficiency group, and high fat, high fructose group. In the curve, although we didn't find a, you can see there is a decrease of phospho-AKT level in both the vitamin D deficiency level and high fat, high fructose level. Similarly, GLUT4 level also it was down. Because our number of sample was only 3, if we put up more sample, we could see much more difference. We get significant value, but with the 3 sample we have done, we're not able to got that changes. Anyway, and TNF alpha, which at the same time we can see a huge change in inflammation, TNF and IL-6, which is clearly seen. It is very interesting that high fat, high fructose diet, they have not able to increase TNF alpha, IL-6, but we have seen a huge increase of TNF alpha, IL-6 in vitamin D deficiency diet. So it's clearly showing that in inflammation also happening in cardiac muscle when we are having a vitamin D deficiency diet. So again, SOD2, which is the mitochondrial superoxide dismutase, one of the important antioxidant, which neutralize superoxide dismutase to make H2O2, and then H2O2 need catalyst to to neutralize to water and oxygens. So we measure SOD2 and catalyst in their protein expression, which was clearly shown that SOD2 was decreased in diabetes group as well as high fat, high fructose group. Similarly, catalase was only decrease in vitamin D deficiency, but we have not see any decrease. Rather, it was increase in high fat, high fructose. So, telling that vitamin D deficiency diet actually causing more changes in heart muscles in terms of inflammation and oxidative stress. So, so vitamin D deficiency induces myocardial insulin resistance coupled with inflammation and oxidative stress. This is the conclusion from this molecular biology data. Then we also wanted to see the role of vitamin D receptor agonist on insulin resistance and cardiac function in high fat, high fructose diet. So this is my second objective. But as I said, if vitamin D deficiency is causing a problem, we've seen that high fat, high fructose diet, they cause metabolic syndrome. Now, if we that model, because we want to see if any diabetic patients come to the clinic and they have cardiac dysfunction, insulin resistance, if you're giving a VDR activator, so whether the patient will work or not. So we have done that study in animal model. So we choose a 4 different groups: control diet, 20 weeks. We're giving a high fat, high fructose diet, as this is a type 2 diabetes model, 20 weeks. We're giving high fat, high fructose with pericalcitol, is a direct vitamin D receptor activator. And we also compare with this study with a standard compound enalapril. It is a AC angiotensin converting enzymes inhibitors, which also have some good effect on diabetic heart. So if you look the data and blood glucose level, as we have seen previously, the high fat, high fructose, they're having increase of blood glucose, but other two, there also increases, but although it's not found significant. Similarly, insulin level was very high. It was high in high fat, high fructose group, but we didn't find a significant decrease of either VDR agonist or enalapril group. Similarly, we didn't find a huge change on HOMA-IR, although we got a change in high fat, high fructose. VDR agonist, there's a decrease, not significant, but enalapril not so any changes. But when we measure intraperitoneal glucose tolerance test, so we can clearly see the top curve is showing that in the left side in this graph, in the top one, the curve as going up and down is showing high fat, high fructose group. Control is the below one, but both the VDR agonist and enalapril, they are in the middle. That's showing that yes, definitely there are some effect of on insulin resistance with the both enalapril as well as pericalcitol. Similarly, when we measure serum triglyceride level as well as serum high density lipoprotein level, so we can see only triglyceride level was significantly down after VDR agonist. We didn't find any change on HDL level. Although we can see the trend, there's an increase of HDL level after VDR agonist, but because they are high standard deviation, so we could not able to any get significance. Again, systolic and diastolic parameters and mean arterial pressure, you can see this is very good data, interesting data, that only vitamin D actually showing significant changes of systolic blood pressure, diastolic blood pressure, and mean arterial pressure. But enalapril, which is a well-known antihypertensive drug, although it has a trend to decrease, but vitamin D was, or this enalapril, this VDR agonist, pericalcitol was much better compared to the enalapril. Then we did the echocardiography study and to look that whether cardiac function has improved with the VDR agonist. So here, first one, interventricular septal diameter thickness as well as left ventricular posterior wall thickness. In both cases, we can see there's an improvement of this thickness of the heart muscles by vitamin D receptor as well as enalapril. There's no change on left ventricular internal diameter, the thickness on both systole and diastole. And when you measure fractional shortening as well as ejection fraction, you can see that then both high fat, high fructose, there is a decrease of fractional shortening and ejection fraction, which is a two important parameter of cardiac function, which was improved, I think, improved better than enalapril after VDR treatment. So pericalcitol able to improve the cardiac function and much better than enalapril group. So overall, if you look the chart, so VDR agonist, they able to decrease insulin resistance, able to improve HOMA-IR, decrease intraperitoneal glucose tolerance test, triglyceride, serum triglyceride level, HDL also increases, systolic blood pressure, diastolic blood pressure, that decreases, mean blood pressure also decreases. When you think about echocardiography point of view, the cardiac function, we can see there's a decrease of interventricular systole diameter and left ventricular posterior wall thickness, as well as we can see there's an improve of fractional shortening and ejection fraction. So overall, this data clearly showing that VDR, vitamin D receptor, could be a very good target for cardiometabolic disease, especially when you're having a diabetes, insulin resistance, as well your cardiac function is deteriorated. So VDR could be an important target. So just to find a mechanistic study, when you go to the in vivo model, whole animals, so there are a lot of other factors are there. Like sometimes you having a lot of hormones, lot of other factors that play important role, and you don't know whether VDR is a real target or not. To confirm that VDR is a real target, so we took H9C2 cells. This is a rat cardiomyoblast cell. Now we knocked down the VDR in H9C2 cells. So now we got two different groups. One is H9C2, which is a VDR is intact. Another is H9C2, where we have a knockdown of VDR. You can see the RTPs here. So most of the we have shown that there's a decrease of VDR vitamin D receptor gene. So we have silenced this gene, and then we measure actually 2 NBDG uptake. 2 NBDG is a it is a dye, is a kind of glucose substrate. But it is attached with one dye. So if you give palmitate, if you treat it with the palmitic acid in the cell, H9C2, they got insulin resistance. So they don't get a lot of glucose uptake. So if you see that palmitate, there's a decrease of glucose uptake in the H9C2 cells. But if you're giving, like pericalcitol, there's an increase of little bit glucose uptake. But the same study in the B graph, you can see we have done with the insulin. So that we control H9C2 cells. If you give insulin, there's an increase of glucose uptake in H9C2 cells. If you have license, even a silence VDR, okay, so again, then it is a decreased glucose uptake to NBDG uptake compared to the control group also. But when you treated VDR silence cell with insulin, we not see any further increase, which you have seen in the in the first two group, control and control plus insulin. You can see there is almost two-fold increase, but we've not seen increase out here when you silence this one. So definitely tell that insulin that increase glucose uptake in cardiac cells through VDR. So there are some crosstalk is there in this signaling pathway, insulin signaling pathway, which I think we need to look more further into animal studies also. So in the bottom, actually, we have also look actually phospho-AKT by immunofluorescence, and also we look GLUT4. So if you give insulin in the second, in the I think these are my last 1 and 2 slides. I hope I can finish in time. So here, if you give insulin in the C group, so there's an increase on phospho-AKT level. You can see the fluorescence has increased. At the same time, in the right side, your GLUT4 translucence is very high. So you can see very bright of this green fluorescence. But if you have knockdown in the last, in the figures, if you knock down VDR and give there is no increase of phospho-AKT. Also, we have not seen any increase of GLUT4. So which telling that if you block or if you inhibit VDR, so insulin will not work in this cardiac cell lines. So it will not activate phospho-AKT, so it will not translocate GLUT4 into the membrane. So vitamin D and its receptor VDR positively regulates myocardial insulin sensitivity. We come to the conclusion from this study. So now coming to the conclusion. So as you've seen, there are a lot of actually study from human to animal and then cell line. So we come up to a conclusion that vitamin D deficiency alone induces insulin resistance and cardiac dysfunction. Is a very important conclusion because who are vitamin D deficiency for a long time, who have vitamin D like several years, 5 years or 10 years, please check it, because if you remain vitamin D deficiency, you get a lot of problems. Sometimes doctors give a treatment of with some other drugs, but you will not respond because you need a simple and need supplements of vitamin D, you'll be completely fine. It happens several times, few cases. We have seen in Delhi, one of the hospital, one patient came with a heart failure, and doctors realized that he has a very low of calcium and vitamin D level in the blood. So doctors not treated for heart failure. He gave a treatment of only vitamin D and calcium, and the patient within a few days remained fine and sent back to the home. So we remember that it is very important, and now we know that how important for cardiac function also vitamin D. So vitamin D receptor activator improves cardiac function in metabolic syndromes with effect comparable to current standard treatment. So whether we need to explore whether vitamin D receptor could be a treatment therapy for heart failure patients. And pericalcital, which is a VDR activator currently used in management of kidney disease, shows a potential for the treatment of cardiac dysfunction in metabolic syndrome. So there is a repositioning, or repurposing of this drug pericalcital for heart failure and also type 2 diabetes. So as the whole data generated by a lot of students in my lab, Dr. Ramu Adela, Dr. Hina Latif, all they completed PhD with me. Dr. Nomin Reddy, without whom I cannot do the human study. Dr. Art Srinivas from CSR IICT, he is the key player for measuring vitamin D in patient sample. Professor S.K. Maulik from AIIMS New Delhi, who is also guiding us different time whenever we are doing the study and looking different parameters. Professor Pralay Chakraborty, who is a cardiologist in Safdarjung Hospital, and who also time to time give a feedback about our work and study. And definitely without that we cannot do is a funding agency, DBT and CSR, they support a lot for this study. And I like to thank you to all of you. I put the pictures. This is our new campus. Is a Naipar Guwahati. Is a a beautiful place. So all of you are requested to invite whenever you visit Guwahati. Please visit us. We having a good research facility, good team, and we have a good animal facility. And thank you very much for giving this opportunity. Thank you.
Thank you, sir. Now the session is open for discussion. I'd like to welcome Ms. Shinti P. to hand over the session, please.
Thank you, sir. It was a very informative speech. So let me ask the questions. First question is, can lack of vitamin D cause brain fog? Can lack of vitamin D cause brain fog?
Okay. So yes, there are studies are there that vitamin D deficiency cause depression and some kind of effect, neurodegeneration. Some studies are there, and sometimes we remember that vitamin D actually vitamin D receptor VDR present in every cell, starting from your smooth muscles, skeletal muscles, monocyte, macrophage. So the importance of VDR is so important. Vitamin D also play important role in immunological function. So brain also, neuron also very much important for this function. And but yes, I don't want to talk much because I don't know much on its direct role or indirect role on brain, but definitely there are studies there.
Okay, sir. Thank you. Next question is the insulin ineffective in a diabetic patient who is vitamin D deficient? Is the insulin ineffective in a diabetic patient who is vitamin D deficient?
Yeah, I think very good questions. Yes, I answer it different way. When you're having a vitamin D deficiency long run, definitely your insulin signaling pathway that actually hampers. So even you give insulin, it will not work. We have shown that when VDR is not active, when VDR we have silence, so insulin is not working. So definitely when you're having a vitamin D deficiency, so insulin signaling path will not work as the way it works. Yes. So you should having a normal vitamin D level in your blood so that insulin work perfectly.
Okay, sir. Next question is there any relationship between vitamin D deficiency and sleep disturbances? Vitamin D
Yes.
deficiency and sleep
Yeah. Sure. I see when I talk about different symptoms of vitamin D deficiency. Okay, most of the cases we get a muscle weakness, fatigues, all these things. Okay, muscle cramp. But it is also very well known that vitamin D deficiency causing a sleep disorder. It is there. So yes, it is reported and it is a very well known fact.
Okay, sir. Another question is, does vitamin D have any role in reducing respiratory tract disease? Any role in reducing respiratory tract disease?
Yeah, now I have to be, so frankly speaking, I have not read any article directly linked to respiratory tract disease, but indirectly I can tell you, as you see that vitamin D has, yes, there are some link I can tell you. Vitamin D actually linked to the linked to the increase of nitric oxide level. So it activate enos. Vitamin D activate enos. So when lungs respiratory
Yes, tell me.
alveoli of this work, you need some relaxation of alveoli when you take a breath or something. So that cases nitric oxide play a very important role. So yes, I believe yes there are role which working through relaxation of blood vessels through nitric oxide. There should be some role. Yes.
Okay, sir. The last question is whether vitamin D toxicity will occur with over sun exposure. Vitamin D toxicity will occur with over sun exposure.
Uh, Again, if you go for over sun, the question is that what is over sun? Like you not go for a whole day in a sunlight. You cannot because you get a lot of problem because your body dehydration will be happening. You cannot stay a long time. But minimum requirement is half an hour in the sunlight. Okay, so if you stay 1 hour, 2 hour, I think people do that. I don't think they got any problem. But I'm telling you, if you go for a vitamin D supplementation long time, even you having a normal vitamin D level, so if it cross 100 nanogram or 120 nanogram per ml level, so it causes a lot of toxicity because most of the cases, as you know, vitamin D linked to the calcium absorptions. So they cause a lot of calcium deposition in different part of the body, including that blood vessels. So blood vessels become rigid. So you get not a good effect of vitamin. So you have to remember that because when you measure in human population, we not seen they cross like from I say it from 10 is the minimum level to 120. So very less 1-2 people may cross because of sunlight. May not. I don't think sunlight can make increase that much. It's not possible. But yes, if you take supplementation, you will get a very high level and toxicity may come.
Okay, sir. One more question came that is related to COVID and vitamin D. It's how strong is the evidence to the correlation between vitamin D and SARS-CoV resistance on the basis of ACE, ACE2 receptors?
So yes, so if you see that in general belief with COVID-19, it is well known that if you take vitamin C, zinc, and vitamin D, these three are very important for improving your immunological function in your body. Okay, so as I said, vitamin D is very much important for monocyte function and even macrophage function. They have the VDI receptor. They need to activate by vitamin D. So you need to keep your normal level of vitamin D so that when you get the infection, you get problems. So your immunity there boost up. So they should not be down so that the initial fighting with the microbes with the our monocyte macrophage should not be impaired. So vitamin D is very much important. And regarding ACE and ACE2, I don't think there is any direct correlation with vitamin D because angiotensin converting enzyme 2, which expression, I don't think it depends on vitamin D. It's not there. But mostly it is for boosting your immunity. Vitamin D is very much helpful.
Okay, that's all, sir, about the questions. Thank you a lot.
Thank you.
Thank you, sir. Thank you, sir. It was very informative. And now we will move on to our valedictory function. And I think most of our guests have joined. Professor Abhay Kumar Pandey, he has joined. Welcome, sir. And I also request all the participants to stay back, and we'll be distributing the feedback forms after 15 to 20 minutes. And I welcome you all to the valedictory function of our 10-day National E-Conference, Current Trends in Life Sciences. I also like to welcome Vasu sir, our Vice Chairman, Hamsa sir.
Okay.
So Sanjay sir, can you please stop your presentation? Okay, thank you, sir. Thank you. Now I'd like to welcome Ms. Shinsi P to moderate this session.
A very warm good afternoon to all. I invite all the honorable personalities, guest speakers, Principal Dr. Naveen Mohan, our advisor and academic director Dr. B.G. Unni, Vice Chairman Vasudevan Master, Director Board Member Hamsa Master, and faculties, students, and all other participants to this occasion of valedictory function, National E-Conference, conducted by Department of Biotechnology and Microbiology on the topic Current Trends in Life Sciences. I welcome you all. First of all, I invite our beloved principal, Dr. Naveen Mohan, for the welcome speech.
Yeah, good afternoon all. We have come to the last phase of our journey, a journey through the wide world of knowledge, ideas, and revelations. Respected Chairman of the meeting, Dr. B.G. Unni, sir. Respected Vice Chairman of the college, Sri Vasudevan, sir. Director of GEMS College, Sri Hamsa Master. And other invited dignitaries, my colleagues, and participants of this well-appreciated webinar. A sense of satisfaction and confidence rules my mind at this moment. I am satisfied that we are able to handle this historic event of our college in a very good way. I'm confident that we are empowered to shoulder more responsibilities in our academic life. Today, we are concluding this great effort. My duty here is to welcome all the dignitaries present here in this concluding ceremony. In the very outset, let me welcome our research and academic director, Dr. B.G. Unni sir, who is a real architect of the whole webinar. It was his scholarly interventions and timely advices that made this event a reality. Sir, literally you have made it. Welcome you, sir, to this validatory function. As we are all aware, the style and attitude of our Vice Chairman, Sri Vasudevan sir, was helpful in various ways in conducting different programs in our college. Welcome, Vasudevan sir. I am indebted to the relentless motivation poured on us by our beloved director of our college, Sri Hamsa Master. He was always encouraging us to make it a great event. Welcome you, sir. I also acknowledge the presence of incredible researchers like Dr. Abhay Pandey from Allahabad Central University and Dr. Pranab Goswami from IIT Guwahati, who made this conference fruitful in every aspect. Welcome you, sir. I also acknowledge the services of my colleagues, especially Dr. Pino, the coordinator of this e-conference, Ms. Radhika R. Krishna, HOD of Microbiology, Ms. Shinsi, HOD of Biotechnology, Mr. Sama, the technical head of this conference, and all the faculties of Microbiology and Biotechnology department. I welcome you all to this concluding session. And last not the least, I welcome all the participants of this 10-day e-conference. We are indebted to you and we are expecting your participation again in future programs we are conducting in future. Thank you very much. Welcome you all. Thank you.
Thank you, Naveen sir. Now I invite our advisor and academic director, Dr. B.G. Unni, for presidential address.
Thank you. Welcome to the speakers. First of all, I've seen Pranab Goswami and Professor Pandey, and I think all are there. Some of them has got a problems for attending that. They have informed me. Very hearty good afternoon to all the speakers from and state central universities of India, chairman, vice chairman, directors of the board, principal, faculties and various disciplines, and all participants, students from India. The 10 days conference is organized by the James Arts and Science College affiliated to the University of Calicut, is one of the best college having distinction at all its level. I could see a highly unique and talented speakers around the country, namely Professor Abhay Kumar Pandey, professor and former head, biochemistry, Allahabad Central University, talked on the topic oxidative stress and associated complications as inaugural speech. Few important points from his lecture, such as free radicals, toxicity, antioxidant oxidation properties, molecular mechanisms, properties of flavonoids, and role of herbal products. Then we have Professor D. Girija, Professor Agricultural Microbiology and former registrar, Kerala Agriculture University, talked about recycling of various waste, forms of waste for composting. They also had Kerala Agriculture University BioBin and its applications, society with respect to waste management and application fabrication of large devices for waste management at a district level and also the state level. Then we had Dr. G. Mallikarjunasamy, Senior Scientist, Department of Forest Pathology, Kerala Forest Research Institute, talked on nursery diseases, types of pathogens, and control measures. Few silent points from his talk about total contribution with respect to the control of nursery diseases and explained in very detail. Also talked about various ornamental and other common plants and their diseases and control, mentioned about various types of fungal, bacterial diseases and their appropriate control mechanism too at farmers level. We have Pranab Goswami, Professor, Biosciences and Biofuel Cell, Indian Institute of Technology, Guwahati, who was speaking on biofuel smell as smart sensing device for advanced healthcare application. Dr. Goswami talked about biocells and biosensors, its potential application and challenges replacing chemical-based cells to biofuel cell in many body functions. His talk is amalgamated with molecular biology, microbiology, chemistry, physics, mathematics, electronics, computer science, and electrical engineering, an ideal example for multidisciplinary approach in research field. Then we have Professor D.K. Jha, Professor and former head Botany Department, University of Guwahati, spoke mainly on bioremediation of coal mine, significant role of microbial consortium for bio and phytobioremediation at lab level and field level. Also talked about the remediation of heavy metal toxicities in the soil too with the help of microbial consortium and their mechanism. Then the Dr. Devdatta Bhattacharya, scientist and assistant director microbiology, Indian Council Medical Research Regional Center, Bhubaneswar, spoke on the very important current topic on COVID-19, the current scenario and way forward. He talked about one of the very important current subject on COVID-19, where we stand now and how we can go further to tackle the situation. He also talked about various types of testing for COVID and the role of ICMR in this regard. He also touched upon the myths and facts about the COVID and related news. Also the current status of COVID vaccine in India and abroad. Dr. Bula Chowdhury, senior scientist, Guwahati Biotech Park in IIT campus, has talked on a very important topic on entrepreneurship through innovation. She talked about the number of innovation ideas converted to startup commonly used in a day-to-day life. Also talked about the number of innovative ideas developed during the COVID pandemic at different parts of the country. He talked about the different types of funding possibilities and also the current infrastructure facilities available at Guwahati Biotech Park. Dr. Prasenjit Manna, Principal Scientist and Ramalinga Swamy Fellow, CSIR, Northeast Institute of Science and Technology, spoken on the topic, the new perspectives of an old vitamin, vitamin K. His talk was mainly correlated the specific role of vitamin K with respect to control mechanism of diabetes type 2 and other complicated factors in the liver at the molecular level. Professor Arun Lahiri Majumdar, INSA Senior Fellow, Bose Institute, Calcutta. He talked about the various environmental factors affecting the plants from the environments and also discussed in details about the abiotic and biotic stress mechanism and the effect of salinity. He explained the entire molecular research to reach the final stage. The way he explained the molecular mechanism so simple and each and every student of life sciences can understand. Dr. Sanjay K. Banerjee, Associate Professor, National Institute of Pharmaceutical Education and Research, Guwahati, spoke on the vitamin D deficiency and cardiometabolic syndrome, a study from bench. He also some of the points, vitamin D and the deficiency related, also related to the cardiovascular diseases, type 1 diabetes, and also correlated the relation between by its molecular level and explained the mechanism too. In fact, all the speakers delivered from 14th September till today on topics that was very much relevant to the lecture. I am sure and confident that all the participants had an excellent opportunities to listen and interact with the learned speakers during the interaction section during the last 10 days. You may also notice that most of the speakers what spoke from different background, starting from CSIR, ICMR, DST, DBT, direct government funded projects. So you all has got some an idea about the research and then the state government funding agencies, state universities, central university. So you overall you got the research atmosphere, how they perform and what sort of facilities they have got and what sort of research idea, how they proceed further. Each and every aspect through these 10 speakers, most of the faculties and students have got knowledge. All the lectures selected by the speakers of their research area were highly informative and very much useful to the faculties and the students. On this last concluding day of the e-conference, I convey my greatest appreciation and very sincere thanks to all the speakers from different parts of India for their constant to be the part of the conference organized jointly by the faculties of microbiology and biotechnology department of GEMS Arts and Science. My special thanks to the entire team of the organizing committee of the e-conference, convener, organized coordinators, and all other supporting team members for their continuous, untiring effort for the successful coordination till the completion of the e-conference. This is the best way to know more details about recent development in science and technology and the way the research is being carried out, to know more at different R&D institutions from the respective speakers. I'm extremely thankful to the chairman, vice chairman, director, and the board of management of the college for their continuous support for all the time. I'm very happy to be the part of the event, and such activities will continue in future too. Thank you very much.
Thank you a lot, sir. Now, I invite Professor Abhay Kumar Pandey, Professor Head Biochemistry of Allahabad Central University, to speak a few words.
Thank you, Dr. Saini. Am I audible?
Yes, sir. Yes, sir.
Yes, sir.
Good afternoon to all of you. At the outset, I thank you all, especially the GEMS fraternity. All of you have put in your best efforts, so you deserve a special thanks. So far, this conference is concerned, National E-Conference on Current Trends in Life Sciences. So I'm sure the inputs of various speakers from diverse parts of the India, from these inputs, the delegates and participants must have got benefit. It was, I think, the program was immensely successful, and I'm thankful to Dr. B.G. Umi, the Dean, Academics and Research, for taking initiatives for organizing this conference. I would like to thank Dr. Mohan, Dr. Finose, Dr. Saini, and all your faculty members from the GEMS College of the Calicut University, and wish all of you good luck. Thank you.
Thank you a lot. Now, I invite Professor Pranab Goswami, Professor of Biosensors and Biofuel Cell at Indian Institute of Technology, Guwahati, Assam, to speak a few words.
Hi, good afternoon to you all. Respected dignitaries in a virtual dais, Dr. Mohan, Dr. Unni, Sri Vasudevan, Sri Hemsaji, Sri Nawazji, Dr. Finos, and all other respected speakers, Dr. Pandey, and Dr. Banerjee I have seen, Dr. Rajalakshmi. So I have seen a galaxy of eminent personalities sitting in this virtual dais. So it reflects the success of this e-conference. Well, the success of this e-conference is obviously due to the eminent speakers and the excellent organizations organized by the organizing committees. But also I have seen another very interesting thing over here, the curiosity of the young minds. When I had delivered the lectures, and in the last lectures also I have seen the questionnaires from the students. Really I appreciate it, because it reflects that speakers could stimulate the interest of the young minds. So, which indicates that this conference is really successful. That means the knowledge of the speakers, eminent personalities, could be pervaded to the young mind of your college and participants. So I need not have to tell more than this to reflect clearly the success of this e-conference very much. Thank you very much.
Thank you.
Thank you a lot, sir. Now I invite Dr. Sanjay K. Banerjee, Associate Professor, National Institute of Pharmaceutical Education and Research, Guwahati, Assam, to speak a few words.
So this is a great initiative from James Arts and Science College. I think we have seen several e-conference, but it is one of the conference which continue 10 days, and you people together you organize so well, and keep a time also like 2:30 to 3:30 is a very important time. Like you finish your morning all official work, what are your classes, all these things. Now 2:30 you can keep yourself for the conference, and you can attend this one, you can ask questions. So timing is also very important. We don't I think none of us is actually affected from our daily activities in the institute. So, and I can see the kind of lecture and topics you have chosen, which will give a exposure to all kind of research. Like mostly who are the attendance are students, maybe MSc, PhDs. They'll get ideas actually, and especially who are the initiator. Like they're thinking that okay, the completion of master degree, they want to start their research, so they'll know, at least they get some sense of different kind of field. So I've not seen a kind of variety of topics you people chosen in any conference. So I think it's a big congratulations to all the team and Professor Uni. I think all efforts you people put together, I think really having a great success. And as Professor Pranab Goswami said, the questions was really good. Even today also, when I was answering the questions, I really I have to take some time to answer. I try to correlate the questions they ask, how to put together and how they link each other. So yes, they have listened very attentively. The way they ask questions, it looks that you are really successful. And what is the main motive of the conference? You have achieved it. So congratulations to the whole team, and thank you very much to make me part of the team. Thanks a lot.
Thank you.
Thank you a lot, sir. Now I invite Professor D. Girija, Professor of Agricultural Microbiology and Registrar, Kerala Agricultural University, to speak a few words.
Good afternoon, everybody. I'm so happy to be present during the valedictory session also. I take this opportunity to congratulate the organizers for arranging this e-conference for 10 days. I found that the topics were very diverse and interesting also, ranging from plant stress to biofuels to COVID-19 pandemic. So it was very interesting. All the topics were very interesting. And I found I also found that the participants were very attentive during the entire sessions. I attended only a very few sessions, but I found that even today's session, they were all very quiet, and they were listening very attentively to the sessions. And just like my previous speakers told, they asked very genuine questions, showing that they have paid much attention to the topics, and they were they were provocated to ask good questions. So I congratulate the team again. I also congratulate the participants for having successfully completed this conference. Thank you so much, and hope that more such activities like organizing e-conferences will come up from your side. Thank you so much.
Thank you.
Thank you a lot, ma'am. Now I invite Dr. Finos A., Assistant Professor in Biochemistry, for valedictory address and vote of thanks.
A very good evening to all the dignitaries, speakers, management committee members, and all the participants in this event. Today, as the coordinator of this event, I feel very much proud to stand before you in this valedictory address. It is indeed my privilege to share a glance of our 10 days National E-Conference current trends in life sciences. And I'm very much well sure that the student community who have listened to the speeches in our event will definitely have benefited a lot. It was indeed a great pleasure to listen to all the speakers during the last 10 days. Indeed, these 10 days have been an exposure of hardcore research happening in our country. And we were fortunate enough to hear that from the experts in their sessions. Next, I like to move on to the Thanksgiving session. First of all, I like to thank our Honorable Chairman, Sri Manjalanguri Ali, for sharing his time during this busy schedule for inaugurating this event. Next, I like to thank Professor Abhay Kumar Pandey, Dr. D. Girija, Dr. G.E. Mallikarjuna Swamy, Professor Pranab Goswami, Professor Dhruva Jha, Dr. Devdatta Bhattacharya, Dr. Bula Chowdhury, Dr. Prasenjit Manna, Professor Arun Lahiri Mazumdar, Dr. Sanjay K. Banerjee for squeezing out their time for delivering this excellent speech. Thank you all once and again. Next, I like to thank the Vice Chairman, Sri Vasudevan Sir, Sri Hamsa Sir, who is the Management Committee member, for their support and encouragement during this program. Next, I like to thank and express my extreme gratitude to Dr. B.G. Unni Sir, who is the advisor as well as director, academics and research. He has been the motivation and the pushing force behind this whole event. Thank you, sir.
Thank you.
Next, I'd like to thank Dr. Naveen Mohan, who is the principal of our GEMS Arts and Science College. He has been motivating us and driving us forward with his tireless and strenuous efforts. Thank you, sir. Next, I like to thank Sri Mohammad Nawaz, who is our vice principal, who couldn't attend this meeting. I also thank him. Next, I like to thank all the undergraduates, postgraduates, research scholars, and faculties from various universities across all over India who participated in this event for the last 10 days. Thank you all. You were purely disciplined and keen in listening to the talks, which has helped us to conduct this event smoothly. Even though I know the time is running out, I like to express my sincere gratitude to my whole team who worked and supported me for the past 10 days. First of all, I like to thank the joint coordinators, Ms. Shinsi and Ms. Radhika, for helping me out to sort out this event. Next, I like to thank Ms. Rasna, Ms. Renjusha, Ms. Shana, Ms. Gina for their wholehearted support during this event. Next, I like to express my sincere gratitude to Mr. Samad, who is the technical head in this event, who has been behind hosting and streaming activities for this past 10 days. I thank you. Next, I thank you once again for all of you to be a part of our National E-Conference for the last 10 days. Thank you and God bless you.
Thank you. Thank you very much.
Thank you, sir. Thank you a lot. Thank you all.
Okay. Over.
Thank you, everybody.
Thank you.
Thank you
Thank
very
you
much.
very much. Namaste.
Okay, thank you, sir.
Namaste,
Thank you.
everyone.
See you very soon, sir.
Yeah. Thank you.
Okay, thanks.
All the participants, please wait. Your feedback forms will be available shortly. Thank you, all the participants. Those who haven't received the certificates, you will receive it shortly. Okay, within 10 days, we'll be providing you all with your certificates, especially pending certificates. Thank you.